Fungi and fungus-like organisms comprise the largest group of pathogens of plants. In this section, we will describe the basic characteristics of these pathogens, how they are organized taxonomically, and how they reproduce on plants and in the environment. This section includes the zygomycetes and ascomycetes. Other subgroups will be covered in the next lecture. There are three major groups of fungus-like organisms that contain pathogens of plants. The largest and most diverse group is the true fungi, shown in the left column. Of these, we will focus on the zygomycetes, ascomycetes, basidiomycetes, and metasporic fungi, which are also called deuteromycetes or fungi imperfecti. These are fungi that reproduce asexually and for which a sexual stage is unknown. The zygomycetes have only a few genera of interest to fruit and vegetable groups, but the ascomycetes, basidiomycetes, and metasporic fungi contain many destructive pathogen species. In the middle column are the stramnaophiles, which include the oomycetes, the only class of interest for this course. The oomycetes were initially considered to be fungi, but based on genetic, morphological, and biochemical characteristics, they are now considered to be more closely related to algae than to fungi. Important oomycete plant pathogens include Phytophthora, Pythium, and the downy mildews. Finally, in the right column are the slime molds. Most are not of consequence to fruit and vegetable pathology, with the exception of the plasmidiophomycetes. Plasmidiophora brassica causes clubroot, a very important disease of crucifers, which will be described in the brassica lectures. In the last slide, we went through a relatively simplified organizational scheme for fungi. Before molecular genetics provided objective clues to relatedness of individuals and groups, fungi and other organisms were classified based on morphology, or what they look like, and their means of reproduction, especially in the ascomycetes and basidiomycetes. This led to confusion since the asexual and sexual states of fungi are morphologically dissimilar, resulting in separate names for the two states. Therefore, a basidiomycete fungus, such as Rhizoctonia solani, has a sexual or perfect state called Thanatophorus cucumis. A movement within the mycology community, called the One Fungus, One Name, proposes to utilize genetic marker data to organize fungi and select a single name. A blog called The Heifel Tip has a post that explains this concept and refers the reader to journal articles supporting it. The link is posted in your reading list for this section. Most fungi produce both vegetative and reproductive structures. The mycelium is the vegetative part of the fungus. Fungi absorb nutrients from the environment through the mycelium, which also acts as a transport network for the fungus. The individual elements or strands of a mycelium are hyphae. Hyphae vary in diameter, color, septation, and branching patterns, among other characteristics. Many true fungi have septate hyphae. The septa are walls that divide the hyphae into separate cellular compartments. Septa are porous, having tiny perforations that allow the cytoplasma, and in some cases organelles, to move from compartment to compartment. True fungi that are aseptate, meaning they don't have septa, are in the zygomycetes, such as Mucor. However, septa are found at the branching points in the hyphae. This is a protective feature that allows the fungus to isolate damaged sections of the mycelium. They also serve to separate vegetative from reproductive forms. In some cases, the morphology of the hyphae is distinctive and can be used as a diagnostic tool. For example, Rhizoctonia solani has very characteristic right angle branching of the hyphae, often with constriction at the branch point, as seen in the first image on this slide. The septae consistently are located at the same distance from branch points. The arrow in the first micrograph on the left points to a septum near a hyphal branch point. In some fungi, including numerous plant pathogens, the hyphae may self-organize into another vegetative structure called a sclerotium. The outer layers harden and melanize, protecting the hyphae inside under unfavorable environmental conditions or lack of a food source. Sclerotia come in various shapes, colors, and sizes depending on the fungus. Sclerotium rolfsi, pictured on the right, produces round, tan to gold-colored sclerotia a few millimeters in diameter, and often are described as looking like mustard seed. Sclerotinia sclerotiorum, which you will hear more of in later lectures, produces irregular-shaped black sclerotia that can exceed 1 to 2 centimeters in length. Sclerotia may germinate vegetatively, producing more mycelium, or may produce a sexual reproductive structure. Most plant pathogenic fungi and fungal-like organisms produce asexual reproductive structures called spores, sometimes termed midospores because they are produced through mitosis. Cnidia is the term commonly used for spores of most true fungi. Sporangiospores and zoospores are reproductive mitospores of the oomycetes. Sporangiospores and zoospores are also found in Plasmodiophormycetes, although the sporangiospore is produced through meiosis. Zygomycetes also form sporangia. These will be described in greater detail later in the course. Chlamydiospores are usually round vegetative structures within hyphae and also serve a protective function. Meiospores are spores formed through sexual reproduction and thus involve meiosis. Ascospores are formed in ascomycetes, often in a sac-like structure called an ascus. Basidiospores are produced on a basidium by fungi in the basidiomycetes. The focus of the remainder of this section will be on zygomycetes and ascomycetes, both of which are true fungi. Zygomycetes are true fungi that are mainly aseptate, although septa are formed to separate vegetative and reproductive structures. They reproduce asexually by spores without flagella in sporangia. Sexual reproduction occurs via two sexually undifferentiated gametangia. These are gametangia that look alike, although conjugation is between plus and negative types. Rhizopus and Mucor are the two most common zygomycete genera among plant pathogens, and both are major post-harvest pathogens. They both produce cream to dark-colored, loose colonies with small black pinheads visible to the unaided eye. Rhizopus is ubiquitous in the environment and is also known as common bread mold, and can be a troublesome lab contaminant as shown in the photo here. This is Rhizopus on summer squash, where you can clearly see the sporangiospores and sporangia. Now we'll move to a much larger group of true fungi that contains many plant pathogenic species, the ascomycetes. Ascomycetes are commonly referred to as cup fungi because of the shape of their sexual structure, also called an ascocarp. In the left photo, you can see the concave cup-like formations within the ascocarp of the morel mushroom. Most edible mushrooms are basidiomycetes, but the morel is an ascomycete, and ascospores are produced in and released from cup-like formations. The photo on the right shows two ascocarps, specifically called apothecia, that have arisen from a sclerosium, indicated by the arrow of Sclerotinia sclerosiorum. In the ascomycetes, hyphae are septate and form the vegetative mycelium. Asexual reproduction is via the formation of conidia, which may be produced in the open on conidiophores or in structures called fruiting bodies, including pycnidia, acervuli, or stroma. Sexual reproduction occurs through the production of an ascocarp. Ascocarps vary greatly in morphology among different genera of ascomycetes. Ascospores, a product of meiosis in the ascocarp, are usually born in an ascus, and since they are a product of meiosis, there are usually 8 ascospores in an ascus. There are a great deal of variation in reproductive structures of the ascomycete fungi. We'll go through the common types of ascocarps, which have taxonomic as well as biological importance. The first, and not very common among plant pathogenic ascomycetes, are the naked asci, produced by genera including Taphrina, which causes peach leaf curl and other diseases. In this case, no ascocarps are formed. Asci and ascospores are produced directly on the leaf surface. The next type of ascocarps are spherical structures enclosing the asci and ascospores. One type is a Cleistothecium, produced by a number of genera, including Penicillium. Ascospores are released when the wall of the Cleistothecium degrades. Chasmothecia looks similar, but ascospores are released when the Chasmothecium bursts. Fungi that cause powdery mildews produce this type of structure. These structures also protect the ascospores from adverse environmental conditions. A perithecium is a mostly enclosed structure but contains a small opening called an ostiole. Perithecia are embedded in plant tissue near the surface so that the ostiole is open to the environment. Asci and ascospores are released from the perithecium through the ostiole. Finally, the apothecium is a common type of ascocarp featuring a cup or disc-shaped structure. The asci and ascospores line the surface of the cup and are released directly into the air under appropriate environmental conditions. For Sclerotinia sclerosiorum, ascospores are ejected into the air when the humidity drops, an ingenious dispersal mechanism. The next and final video for the introductory lecture will cover basidiomycetes, oomycetes, and plasmodiphoromycetes.